Just wanted to finish up section 5.2 here. We were talking about the structure of virions, the structure of a virus, and the last structure that I wanted to just elaborate on was the importance of spikes. And a lot of these spikes are proteins. Remember, some virions have them, some don't. But one of the things some of the spikes do is they bond to receptors on host cells. So host cells, every one of our cells have different kinds of proteins on the surface that identify them as liver cells or skin cells or respiratory cells. And so virions that have spikes, sometimes these spikes actually recognize those receptors. And so this is showing you an example of a virus attaching to a host cell to those using the spike proteins, and it brings it into the host cell. The other things notable about spikes, there's some other important things that we're not going to go into, but one of the notable things is because they're proteins and we generally like to make vaccines against proteins, a lot of times these spikes are used for targets for vaccines. So we make antibodies against these spike proteins to protect ourselves. So when you hear about like the H1N1 virus, which is an influenza virus, it's describing the kind of spikes that the virus has the H1 type and the N1 type. So, so spikes can be pretty important in the structure of virions. The last thing I wanted to talk about when we talked about acellular pathogens is our prions. So we're out of viruses right now, but there is another pathogen that can cause infectious disease that is non-living, is acellular. And these things are called prions. And prion got its name from proteinaceous infectious particles, and someone just shortened it as prion. So prions are not cells, prions are not viruses, and prions are their own category of infectious agent. Now, these are not very common. All of us will have bacterial and viral infections. Probably most of us will never have a prion infection. These prions are really weird because they are only protein. They don't have nucleic acid. And if you remember about your nucleic acids, your DNA and RNA, they contain the instructions on how to make new cells, new proteins, new cell parts. Prions don't have DNA or RNA, so it was kind of one of those things that when they were discovered, particularly starting in the 1980s, that people were saying, well, they can't possibly be infectious. They don't have DNA or RNA. But we found out that they can multiply themselves in a completely unique way. The other thing about these prion proteins, you might remember that when you heat proteins, they denature. When you put them in acid or expose them to radiation, things like that, they denature and they lose their function. But prion proteins are exceptionally chemically stable. And you can cook them, you can wash them, boil them, anything, add disinfectants, and they don't inactivate. So they're incredibly stable, and that also creates some of the complications when you're dealing with the prion disease. I have a sheep here that has a prion disease called scrapie, which I'm going to talk about. So how does someone know if they have a prion disease? The signs and symptoms of prion disease first can occur with kind of the changes, and these can happen in humans or some certain animals that I'll mention in a minute. But all prion diseases cause confusion, memory loss, personality changes, uncoordinated movements, and they're ultimately fatal. And the reason why these prion diseases cause these kind of things is they affect the brain. And so if you took A&P, you might remember seeing brain tissue, and it's nice and should have like all sorts of nice tissue there, and you see some neurons, but you shouldn't see these big holes. And so having a prion infection causes these holes in the brain and in brain tissue, and that causes these characteristics that will be typical of a damaged brain. So all of these will happen in any living animal that gets a prion disease. And so because of this, we call prion diseases transmissible, meaning you can pass from organism to organism, spongiform. You notice the brain kind of looks like a sponge under microscopic examination. Encephalopathy. Encephal refers to brain. Pathy means disease. So we would just call this like a contagious spongy brain disease. And you can see it, prion diseases shortened as TSE or sometimes just SE. The most common TSE or prion disease in humans is Creutzfeldt-Jakob disease. And they're showing you a picture right here of a brain of a person that has that. This is obviously a deceased person versus a control of a normal brain. And notice how much smaller it is, how much bigger the spaces are. So this is this spongy brain that's kind of collapsing on itself. And you can imagine that would be devastating to a patient. So how do prions infect host cells? I put this diagram on your study guide, and I hope you would use it and practice with it. So I could give you a lot more complicated diagram. I'm giving you the simple version. So the idea is, when we have a viral infection or a bacterial infection, the virus or the bacterium has its own DNA or RNA, and it can multiply. It can make copies of itself. Prions are just proteins. So it turns out, so in this diagram, these red spiky things are prion proteins, and these green ones are healthy proteins. So everybody's brain, most mammals, maybe all mammals, I'm not sure, have these normal brain proteins, and we're just going to call them PRPC. These are the normal brain proteins. And so all of our brains have these. If your brain gets exposed to a prion protein, and they're calling these PRPSC, you don't have to remember that terminology. Just recognize that we have normal brain proteins. If they're exposed to this abnormal prion protein, what the abnormal prion protein does is it takes this specific type of normal brain proteins and it refolds them to look like prion proteins. So think of the zombie apocalypse. A zombie walks up to somebody. Okay, I'm not an expert at zombies, but I've seen that zombies walk up to people and you do whatever. I don't know if you bite them or you touch them or whatever, but you turn that person into a zombie. And that's what these prion proteins are doing. The prion proteins are taking perfectly normal brain proteins and turning them into these prion proteins. And these prion proteins start to accumulate and they cause this pathology in the brain. That is the really simple story of this. Okay, so you don't need DNA or RNA to copy the prions. The prions are actually making new prions out of healthy brain proteins. And you get this spongy brain disease, that transmissible spongiform encephalopathy. What kinds of animals get prion diseases? The first disease we were ever aware of, we've known about this disease for last time I saw, like 300 years or more. There have been writings available to talk about this disease called scrapie in sheep. And scrapie is called scrapie because a sheep will stand next to a tree or a fence or whatever and scrape itself. That's what it's doing right there. It's scraping its wool off. It's not acting normally. It's not walking normally. It's not behaving normally. And this disease has been around for a long time. It's not a new disease, even though our understanding of prions is relatively new. But scrapie's been around a long time. There's other diseases. Apparently cats can get a prion disease. Mink can get a prion disease. But probably the other ones you've heard about, you've probably heard about chronic wasting disease, especially if you're a hunter, in deer, elk, moose, reindeer. And this is a disease where both in wild and animals in captivity can get this disease, and they turn out, they lose weight, they're mentally not able to care for themselves, they're just not acting normal. When it really got in the limelight, we were hearing about bovine spongiform encephalopathy, BSE, also called mad cow disease, where domesticated cattle were getting a prion disease. So you've heard of some of these diseases, whether you realize they were caused by prions or not. I know a lot of people are interested in chronic wasting disease, particularly because they're hunters and they want to eat deer that they catch in the wild. And this is a map from, I think made in 2025, so fairly a good recent current map on where chronic wasting disease is in the US. And you can see that it says these yellow ones are captive facilities. The gray marks are where they've found chronic wasting disease in free-ranging populations. New York State is pretty good. Pennsylvania has detected a lot more. It doesn't mean this is in really high numbers. It just means that it has been detected there before. You can see that out west, there's a lot more territory. This is primarily in elk where they're finding more prion diseases. So this chronic wasting disease is found throughout the U.S. and some states have methods that they're surveying this for distribution, trying to just see how common it is. Now, we've never known anybody to get a prion disease from eating a deer with the prion disease, but it's something to keep an eye on. So how would you get a prion disease? Here's a table from your book, and here's some of the common prion diseases in humans. The primary prion disease in humans is called CJD, Creutzfeldt-Jakob disease. And it turns out that the most common way to get CJD is there's just a mutation that people get. And sometimes that mutation is most of the time, that mutation is just spontaneous. So as somebody's developing in utero, they spontaneously get a genetic mutation where they can develop CJD. Now, CJD doesn't show up in humans until they're usually about in their 60s or older. So this happens late onset, about at the age of 68 on average. There's about 200, I've heard up to 300, depending on what statistics I look at, about 200 or 300 cases a year in the US. And if you get this, you die within months. So this is familial, meaning it's inherited. You can inherit it. A small percentage of people inherit it. Most people have a spontaneous mutation where they get this. But this is a rare disease for 200 cases a year in the US. The less common ways to get it is actually one of the first prion diseases we ever knew about in humans that was in, I think it was New Guinea, where people were eating, doing ritualistic cannibalism. People, there was a practice in some local people that when somebody in your family died, it was an honor to be able to eat their brain. And they found that they were getting this disease called Kuru, and they connected it to eating the brains of people infected with a prion disease. So that doesn't occur anymore because they stopped that kind of ritualistic cannibalism, but that's possible. Another way that people can get CJD, which is again, not very common because now we take this into account, is some people have gotten prion diseases from contaminated instruments from surgery. There's a well-documented case where a surgeon was working on doing surgery on a brain with a person with a prion disease, with CJD. They cut themselves and they acquired that disease. There have been cornea transplants. The cornea is the outer layer of your eye, of your eyeball. People have gotten prion diseases from cornea transplants from patients that had cornea, that had prion diseases. So again, these are really common, really rare because we are aware that this can happen and there are precautions that are taken. And these are rare diseases anyway. There's this prion disease called fatal familial insomnia. Again, it's a mutation that can happen in your genome and you die because you can't sleep. And it's called familial because it runs in families. It's genetic. The disease I wanted to to talk about as far as humans, that's kind of was in the news for a while is this variant CJD. So the variant CJD people got from eating contaminant cattle products from cows that had mad cow disease. So the variant CJD is the most notable one that people got from eating cattle products, from eating beef from cows with mad cow disease. And this only happened between 1995 and 2014. Actually, I think the last person to die from this, I saw a number that said 2016, but somewhere around there. People generally died within 14 months, and there were 177 cases. And what was happening is these people that were getting this were young. And so, like the typical CJD, these people were generally older. So these people took longer to die, the variant CJD folks, and they got it at a younger age. And so people started saying, what's going on? And again, look at the time frame, 1995 to roughly 2014. What's that relationship between CJD, the human prion disease, and BSE, which is also called mad cow disease? So here I am, I'm talking about variant CJD and bovine spongiform encephalopathy, BSE, also called mad cow disease. So again, this was first observed in 1986 in the United Kingdom. It peaked in 1993. So there was a disease called mad cow disease. During this time, there was an epidemic going on. In fact, here's the graph. Here's the year where these are the mad cow disease cases. And this was huge in the news. So here we had these cases of mad cow disease. Lots of cows, particularly in the UK, were getting this disease. And what they found is years later, after this peak of mad cow disease, we started seeing these cases of this variant CJD in humans. And so what they figured was people were getting this disease from eating cows with mad cow disease. Now, the number of people that ate beef during this time was much larger than the number of people that died. Eventually, 177 or 178 people, somewhere around there, ended up dying of this variant CJD. It's not an easy disease to get because probably a lot more people were exposed to it. So we don't completely understand everything about this disease, but scientists figured out that people got this variant CJD from eating beef from cows with mad cow disease. Now, how this whole thing happened is what people used to do is, you have a giant herd of cattle. And then there's other people with a giant herd of sheep. And if a few sheep get scrapey or a few sheep die, here you've got a big farm, a factory farm, and a couple of your sheep die, instead of burying them, you grind them up and you put them in the feed for cattle because there's a lot of protein. Well, what scientists think happened in this 1986 epidemic was some sheep died of scrapie. They were fed to cows, and cows got a version of the prion disease that we call mad cow disease. Mad cow disease was not ever recorded before this. So again, we think cows in this 80s and 90s got in the UK got mad cow disease from eating sheep that had scrapie. They ground them up and they put them in their food. And then they believed that humans that ate some of the humans that ate these cows that had mad cow disease got variant CJD. So their brains got this spongiform encephalopathy, and they died of this disease. So, and that's the pattern that we saw. So what did we do? There's now all sorts of regulations all over the world about not feeding. When a sheep dies, you are not allowed to put it into cattle feed. You have to destroy the sheep in another way. So you don't feed the cows dead sheep. And there's screenings in different ways to test cows for mad cow disease. I know one of the things they do in the US is if a cow arrives to a slaughterhouse and it can't stand up, it's not allowed to be slaughtered. And there's other tests you can do. It's not an easy disease to test for, but fortunately, it's a very rare disease. So we take precautions. And so variant CJD people got from eating cows with mad cow disease. That's the story for prions. And I'm going to end there.